I'm going to discuss the term which is centrifugation. How to define centrifugation? We can define centrifugation as the process that involves the use of the centrifugal force for the sedimentation of mixtures with a centrifuge. What's wrong with the components in a centrifuge? More dense components in a mixture migrate away from the axis of centrifuge, while less dense components migrate towards the axis. As you can see in the diagram, the horizontal arrow shows centrifugation time, while the vertical arrow shows the centrifugal force. Products of centrifugation. We obtain two products after centrifugation. The one is pellet, the other is supernatant. Pellet is in solid form while the supernatant is in liquid form. This is the product we obtain after centrifugation. Pellet are the precipitate that gather on the bottom of the tube, and the remaining solution, which is known as supernatant, is a liquid that is decanted from the tube without disturbing the precipitate. This is a centrifuge. It is a device that accelerates gravitational separation of substances that differ significantly in their masses. Next, we are going to discuss centrifuge uses centrifugal force, that is G force, to isolate suspended particles from their surrounding medium. What are the components of centrifuge? There are many components of centrifuge. The one is rotor, number two is drive shaft, then centrifuge chamber, power switch, timer, speed control, protective shield, refrigerator, and audio and visible alarms. These are all the components in the centrifuge. Centrifugal process. It is a process using a rotating machine to generate centrifugal force to separate substances of different densities. Centrifuge separates molecules according to their masses. As you can see in that diagram, the result of centrifugation, small sized DNA molecules are at the bottom of the tube, while medium sized DNA molecule in the mid, while the large sized DNA molecule is at the bottom. This is the result we obtained after the centrifugational process. What is RCF? RCF is relative centrifugal force. It is used for the determination of speed of centrifuge. What is the formula of RCF? RCF is equal to empirical factor into R into N, where R is the horizontal distance or radius, N is the speed of rotation in RPM, This is the diagram showing reduction of relative centrifugal force influences cell number and growth factor released with injectable PRF based matrices. You can see the results when we provide high RCF, medium RCF and low RCF. Types of centrifuges. Number 1 is high-speed centrifuge. High-speed centrifuge spin at a speed of 15,000 to 20,000 rpm and is usually have large sample capacity. This is the diagram of high-speed centrifuge. Next is microcentrifuge. Microcentrifuge spin at 12,000 to 13,000 rpm and take up to 2 ml volume. It is small enough to fit at the tabletop. You can see its diagram. The last one is ultracentrifuge. Ultracentrifuge spin at 65,000 rpm and it is able to isolate the particles like ribosomes, proteins and viruses. it is very much expensive. This is the diagram of ultracentrifuge. Types of centrifugation. First type is preparative centrifugation and the other is analytical centrifugation. Firstly, we can see the further types of preparative centrifugation. One is differential centrifugation and the other is density gradient centrifugation. Density gradient centrifugation is further divided into two types. One is rate zonal density gradient centrifugation and the other is isopycnic density gradient centrifugation. Differential centrifugation. In differential centrifugation, particles are separated on the basis of their sizes. You can see the largest size particles settle down faster as compared to smaller size particles, and the smaller size particles settles at the end. Density gradient centrifugation. In density gradient centrifugation, particles are separated on the basis of their densities. You can see its diagram. Analytical centrifugation. Analytical centrifugation measures the properties of sedimenting particles like molecular weight or sedimenting coefficient. What is the importance of centrifugation? Centrifugation plays its role in clinical laboratory, pharmaceutical companies, biological research, commercial uses, and astronautics and aeronautics companies. This is all about centrifugation. Thank you so much.